Hi everyone, my name is Hannah. I'm a graduate student in the Lamison Lab at MIT in the bio department. I'd like to thank the organizers so much for inviting me to get to share my work with you all, and thank you all very much for being here. Our lab studies a tick-transmitted bacterial pathogen. These are going to be transmitted normally between ticks and small mammals in the environment, but they can be passed on to people. And all of you are living in New England, so I'm sure you're very familiar with this specific example. This is Borrelia. It's the causative agent of Lyme disease. And our lab studies a different tick-borne bacterial pathogen. They're called spotted fever rickettsia species. And as you might guess from the name, they cause what are called spotted fevers. While Lyme disease is the most common tick-borne disease in the US, these spotted fevers are some of the most deadly, not just in the US, but in the entire world. And they can have an untreated fatality rate of up to 50%, and even with treatment are still quite deadly. And if you haven't heard of them yet, that might change because the tick vector that carries these bacteria is moving northward due to climate change. And within the past few years, these bacteria have been isolated from ticks as far north as New Hampshire and Connecticut. So what actually happens to you if you are bitten by a tick that has these bacteria? The bacteria are going to travel through your bloodstream from the site where you've been bitten, and they're actually going to target the cells that are lining your blood vessels. These are called endothelial cells. And these bacteria can actually adhere to and go inside of these endothelial cells, and they'll start to grow and divide. And as they do, they're going to cause damage to this tissue. This is going to cause your blood vessels to become leaky, and that gives rise to the characteristic spotted rash that you get with a spotted fever. As you might guess, having your blood vessels be leaky is not a good thing. It's going to cause your blood volume to drop, which will lead to a drop in blood pressure, and that can very quickly lead to multi-organ failure, which is what's ultimately going to kill you. And part of the reason that this is so deadly is we have a very limited understanding of the life cycle of these bacteria, which makes it very hard to come up with treatments. And I just want to hone in on the fact that these bacteria are inside the cells in your body. And very much like a virus, these bacteria actually have to go inside of your cells in order to hijack the machinery that they need to grow and replicate and survive. They're what we call obligate intracellular pathogens. And because of this, they have evolved all sorts of really crazy ways that they can hijack our cell machinery and use it for their own nefarious purposes. And the goal of the Lamison Lab is to really understand how these bacteria can do this and how we can better understand that to help treat this disease. A particularly crazy example of how these bacteria can interact with our cells is they're some of the only ones that are known to be able to go into the nuclei of the cells that they invade. And this is pretty wild. If you remember from the last time you took biology, your nucleus is where your genetic information is kept. And so your cell is normally very careful about what is allowed into this compartment, into your nucleus, and even has this entire envelope, the nuclear envelope, that surrounds your genetic material and helps to keep it safe and separate from the rest of the cell. And our lab does a lot of fluorescence microscopy, and this project really started because I started seeing examples of things like this. So these are human cell nuclei, where you can see the nuclear envelope in cyan, and there are these bacteria in magenta with those green tails, and they are swimming in circles with your genetic information around the nucleus. And this is really crazy, and the more that I stared at it, the more I just wondered, how can something this big be going into this nucleus, and why would it want to be here? There we go. And I had a hypothesis, which is that these bacteria could actively get into these nuclei by pushing their way into this membrane. And so it would look something like this. And we turned to one of the most powerful techniques we have in cell biology to answer this. It's called live microscopy. And this just enables us to look and see if we can catch these bacteria in the act of actually getting into nuclei to see how they can do it. And to achieve this, we took cells that had fluorescently labeled membranes. In this case, we labeled a lot of the different membranes in the cell, but also the nuclear envelope. And then we infected them with different colored fluorescent bacteria so we could look and see what was going on. And the image that I submitted for this contest was actually a snapshot in time from one of the first movies that I took to try to figure out how these bacteria were getting into nuclei. And what you can see here are the outlines of cells, just to orient you. So you can see there are a lot of membranes labeled. The nuclear envelope is where this new dotted white line is. And so the nucleus is the black circle in the middle, and you can see these examples of these really large protrusions with many magenta bacteria inside. And what we're looking at here is a single slice through these cells. Cells are complex 3D objects. And so you can imagine if you looked along the side and then you took that slice off and turned it. That is what we are seeing here. And I want to actually show you the movie that I took that helped us to figure out how these bacteria get into the nucleus. And so to do that, we're going to focus in on this cell, and we're going to rewind the movie back to the start, and I'm going to play it. So this is the same cell. We've just gone back all the way to the beginning. The nuclear envelope is again where that dotted line is, and you can see all of those bacteria inside of that nuclear protrusion. And I'm going to go ahead and play this movie. So you can see that the bacteria are really trying hard to get into this nucleus. They're pushing on this membrane. And here it will freeze, so that's where I took the snapshot. And shortly after this, the bacteria actually broke out of that membrane and became free in the nucleus. And so here are some examples of these bacteria. They're now in the nucleus where the DNA is swimming around. And the movie actually didn't end here. So if you keep watching where the red arrow is, something really surprising happened that we weren't expecting. There we go. So now what you're seeing is these bacteria are actually leaving the nucleus. They just got in and they turned right around and they started pushing their way back out, back to the cytoplasm. And so what I've told you about are these bacteria, these rickettsia. They're these very understudied and deadly tick pathogens. They invade the cells in our body in order to survive. And it turns out they can actually go in and out of the nuclei within our cells too. And this is very unique to them. And so the rest of my PhD has been really focused on understanding how these protrusions can be formed by these bacteria and how our cells are able to survive this very violent looking process. And with that, I would like to thank you all. I would like to thank the members of my lab for their support and everybody who's contributed reagents and technical advice and very important funding for us to be able to do this work. Thank you.